subject. Discuss a little bit about the immune response to a viral infection. Frankly, it's really true for most infections, not just viruses, but they're the ones that get all of the credit for the immune system. And how we detect antibodies, antigens. We've already kind of gone over a little bit about some of these, right? In some of the other lectures.
You're in it
Ah,
too.
ah.
I'm just trying to figure out why it won't focus.
I get my better shot. I describe the antigen detection, quantification, methods to monitor disease. Most of that's actually going to come in some of the other lectures about specific pathogens. But I'll briefly go over a couple of methods that we have for monitoring the disease. So, what are the major issues with detecting viruses? Well, the big one is they don't grow like bacteria. We can't just put it on a sheep blood agar and fancy agar. Have a good day for the evening and come back tomorrow and something's better. Right? The only way to actually grow them is with a tissue culture, also called cell culture. In reality, the labs that you're probably going to, certainly ours here at SUMA, we don't do that. It is labor intensive. Cost prohibitive in many cases. Most people are no longer trained properly to do it. It takes a lot of experience to really be able to do cell cultures for viruses. Looking at cytopathic effects, honestly, I would not even be very good at that. You just say, hey, it's normal versus not, but beyond that, pretty difficult to tell. Certainly, I don't think anybody really wants to be playing around with Ebola virus all that much. So, tissue culture is actually very dangerous. Current pandemic, previous pandemic, I guess now. Most laboratories that were still doing viral culture actually discontinued them initially for fear of what the SARS-CoV-2 was. Right? They didn't know much about it at the very beginning, so it was kind of a scary thing. So everybody discontinued their virus cultures. And certainly there's more dangerous viruses out there that you don't really want to be intentionally creating high titers of. And so for virology, serology is the most common. It's kind of being supplanted in it is being supplanted in large part by molecular-based techniques, right? So PCR and nucleic acid amplification techniques are largely supplanting a lot of the serology-based techniques, but many of these are still used today and still valuable. Certainly later in the course of infection when virus detection by nucleic acid detection is not necessarily so successful. So these are still important techniques to know. With a viral infection, there's obviously an incubation stage where you don't even know you got it. The symptoms are absent. The acute stage is what we all realize as being sick. And so it's not the virus itself usually. It's your immune system actually responding to the virus that makes you ill. In the convalescent stage, the virus has been eliminated. You may still have some symptoms, but effectively you are getting better. And every virus is going to be a little bit different. The process is based upon the host's humoral response or antibody response in large part. Immune system, the thymus and bone marrow are the primary organs, with secondary lymphoid organs also playing a role. Deficiencies in one or more of these things land you a one-way ticket to the National Institutes of Health Clinical Center where they can study you. That is where I did my fellowship training, and I can tell you everybody there is an actual hospital, but it's a research hospital, so you have to be on some sort of a protocol. Pretty much everybody there either didn't have an immune system or there was a serious defect in the immune system. And so Your immune system is incredibly important, and there's still a lot about it that we don't truly understand. Um, so, not a place, it's a fun place to work, not a place I'd really want to have to be a patient of. The immune system broken down into the innate, or natural immune system, and the adaptive or acquired immune system. And the innate version is exactly what it is. It's innate habit. These are your external defenses primarily. Right? What's the biggest organ in the body? It's the skin. Right? That's your primary defense against pathogens. If we didn't have skin, we'd all be dead. If not because everything would kind of fall all over the place, but also because we'd be infected with a bunch of pathogens. Okay, so it's your best defense mechanism, and it's innate because it doesn't really care what the pathogen is. Right? That's the big difference between innate and adaptive. Innate doesn't care what the pathogen happens to be. Your adaptive does care. We already mentioned skin, but your mucosal membranes also play a role as your external defense. Internally, we have a bunch of things, including complements. Cellular defense includes phagocytic cells as well as the natural killer cells, which kill things. Right? They're going to directly attack pathogens and infect the cells discreetly. The adaptive immune response, though, is generally what we care about most. It's going to be targeted to specific diseases. It's also what we get to use in the laboratory to help diagnose infections. And so the humoral part of the adaptive immune response is mediated through B cells. Antibody production comes from these guys. We've all, I'm sure, have heard of antibodies and of all the types. The primary ones, IgM and IgG. Ones we deal with most, IgA. Occasionally, some tests include IgA as part of them, but mostly you're going to be working with IgG and IgM. These will generally give you longer term immunity. Like these guys, this is what prevents you from getting sick a second time exposed to a virus. Typically, with the first response, you're going to get IgM, followed by IgG. Your secondary interaction with the virus, you'll get a little bit of IgM, but predominantly a nice strong IgG production, again, helping to either protect you from getting sick or at least keeping the symptoms very mild. Immunoglobulins look like little Ys. Two binding sites here. Okay. IgG is just a single molecule with two binding sites. IgM is 5 molecules attached, 10 binding sites, and IgA looks like that with 2 little Ys going on. These guys are responsible for blocking infectivity of the virus, right? So neutralizing the pathogen. And generally think of these guys against viruses. In theory, they will help with any infection, but most bacteria don't really elucidate a strong IgM, IgG response, or they're able to evade the IgM and IgG response pretty well. So typically we think about viruses. IgA is found primarily in the mucosal membranes, so generally not used most of the time for diagnostics, though they serve important for prevention of infection, but we're not usually doing serology of the nose. The cell-mediated immune response are your T cells. And so humoral are your B cells, cell-mediated are the T cells. CD4, T helper cells, and CD8 T cytotoxic cells. I think the CD8 cells are the ones we tend to think about most in terms of actually functioning, because those are the ones that are doing the attacking and killing. CD4 are important, particularly if you're talking about HIV. So again, mentioned already that these guys are going to kill the virus. particularly the cells that have been infected by the virus. Right? They recognize pathogen patterns, antigens that are presented on the surface of cells that are derived from the virus or other pathogen. They get chewed up, expressed on the outside of the cells. Antibodies move around, recognize it, say, hey, we have a problem. Then the CD8 cells will come around. They also recognize those pathogen antigens. Once they do, it'll induce their killing capability, hopefully stopping the virus or the pathogen from spreading. Again, globulins. I already mentioned the antibodies derived from surface components of intact cells, as well as disrupted virions. And many cell types are able to chew up pathogens and present them. They're called professional presenter cells, professional phagocytic cells, different names depending on what textbook you read. Right. Right. So those same antigens are what we use to create vaccines. And so what is the influenza vaccine? Somebody grew the influenza virus to create the antigens, usually in a killed state or otherwise non-infectious state. We inject it into you. Your cells recognize it and create B cells to create the antibodies. It's no different than being infected by the virus, except you don't get sick. Obviously, you may have an immune response. That's actually a good thing. Like with COVID, everybody, I was sick for 40 days and 40 nights. That was horrible. That's actually a good thing. Your immune system's doing something. You don't have to go to the NIH if you got sick after your vaccine. Right? Your immune system's functioning. I mentioned IgM comes first. Most of the time, there are some viruses like SARS-CoV-2, IgM, and IgG end up being produced kind of at the same time or very close to each other. But usually IgM shows up first for most infections. It's detectable sometime during the acute phase. If there is a lag, your cells don't magically go, oops, virus, IgM, here we go. Right, it does usually take a little bit of time, usually a few days, up to a week or so. But they'll be present during the acute phase of the illness. Okay. Diagnostically, we don't frequently use IgM, at least by itself. If we do, there are certain viruses, certain things like mumps, measles, where we don't expect to be infected, right? It's not a very common infection. So detecting IgM in a patient is actually important and diagnostic of the disease. But most of the time, we don't often use IgM by itself. But when we do, it's usually indicating a recent illness. It could be acute phase. You could be getting better. Just depends on the virus. Again, IgM, usually short-lived, but can last for a long time, depending on the pathogen. Okay. Produced in high amounts initially, but then you don't normally see a lot of IgM in the future. It goes away. Your secondary infection with the pathogen, you'll get a little bump of IgM, but very quickly you get to IgG. At some point during the infection, your cells go, oh, we need to get some IgG going on here. It's the better antibody for protecting you. So they do a class switching to IgG from IgM. And there's an entire science, an entire class that you can take if you want to learn about how that happens. It's a very complicated process. IgG is not going to be detected during the acute phase usually, but it is more stable and long-lived than IgM. Typically with IgG, we're not necessarily looking for it being present or absent. We actually want to get titers. We want to know how much IgG is there. That'll help distinguish between a more recent infection and a longer infection. But in general, when we're doing serology, true diagnostic with serology usually involves IgM testing, or sorry, IgG testing in the acute and convalescent stage. So you need both. And what you're looking for is a fourfold increase in the titer of IgG in the convalescent stage compared to the acute phase, right? We shouldn't have much, if any, IgG acutely. We should have a boatload of it in the convalescent stage. Right? Given that, what do you think the biggest limitation of serology is going to be in diagnosing a patient?
You don't know until they're getting better.
Right. IgG, somebody says, oh, I want to do serology testing on this. Patient just came up with symptoms. IgG is not worth anything. Right? IgG does not diagnose an acutely ill patient. It has to wait until the convalescent stage where it's actually being produced, which means the patient's either better or dead, not overly healthy. So that is a huge limitation on IgG, especially in diseases like COVID, where you're sick initially and then you get better. Right? If you're waiting for IgG, you've already spread it to 100 million people. This diagram of what the production is going to look like. So again, early IgM is going to pop up. You'll eventually get your IgG coming. As IgM decreases, eventually the IgG is going to go down. Sometimes it'll go to 0, or essentially not detectable. Sometimes it'll be detectable. It just depends on the virus. Your secondary infection or reinfection occurs. You might see a little bump of IgM, but you're quickly going to see very high levels of IgG. Right? And that's going to be what protects you again from getting the disease. You may not even know you've been infected again, or at least make your symptoms mild because it's able to control the virus pretty quickly. Seroconversion is the term for suddenly seeing a bunch of IgG. So that's, as I mentioned, a fourfold increase in the IgG between the acute and convalescent stages is considered seroconversion and diagnostic infection. Serology is just the study of antigen-antibody interactions. Most of the time, we're going to be using blood of some sort, either whole blood, serum, or plasma, depending on the test. It is possible to do serology on other specimen types. Occasionally, you'll see CSF looking for antibodies. Most of the time, if you're looking for antibodies, it's going to come out of blood. If you're going to do serology on other specimen types, most of the time, it's going to be antigen detection. Okay? So with serology, we could either look for the antibodies or we can look for the antigen. It's still serology because we're looking at antibody-antigen interactions. It just depends on which one we're controlling and which one we're actually looking for. Titers are largely what we use to measure. This is one of the monitoring methods we have to see how much antibody or antigen is present in the patient. These are usually twofold dilutions, like classically 1, 2, 4, 8, 16, so on. It does not have to be that way. That's the normal convention because it's easy to dilute by hand. Right? But some tests will use a slightly different dilution factor. Others will start out with something. So if you see the cryptococcal antigen test that we do here at SUMA, it's actually a twofold dilution, but it starts with 1 to 1. We do a 5-fold dilution to 1 to 5, and then go through the twofold dilution. So it's really 1, 5, 10, 20, 40, 80, and so on. Again, it does not really matter. You're looking at the titer. The more dilute you can be, the bigger that number after the colon is, the more sample or more pathogen or antibody was present in the original sample. So 1 to 32, dilution means there was more in the original sample than if it was 1:4. And where are we using these? Predominantly IgM. You don't normally do titers on, or sorry, IgM, IgG. You don't normally do titers on IgM, but IgG is very good. Okay. Yeah, you need the acute and the convalescent stage for full increase. Some of the methods talked about particle agglutination in the past with strep typing, right? The agglutination method with latex beads. You can do the same thing for other pathogens and other animals. Okay, so using a particle, some sort of latex bead, that is attached to either an antigen or an antibody, and then you can detect the converse in the patient's sample. So if you want to detect antibodies, you put the antigen on the latex bead, mix them together. And the antibodies are going to bind to a bunch of beads, and eventually you make a big, gigantic mess that you can see on the card. Alternatively, if you want to detect the antigen, you just put a known antibody on the latex bead and look for the same thing. So said you've already seen it with strep typing. It's the same concept. Nice free flowing solutions here. So no agglutination, agglutination here in A. Favorite strep group A. But again, we can do this for any antibody antigen interaction that a test has been made for. Hemagglutination inhibition is the competition between the red blood cells and the virus with antibody. Right? Here we need to have a virus that can do that has hemagglutination properties. Influenza is the most common, has a hemagglutinin protein. Right? And here we're looking for the antibodies to be able to stop that from happening. Okay, so we're actually not going to be looking for the virus in this test. We're going to be looking to see if the patient has the antibodies. But we're going to use one of these pathogens. Again, influenza is the classic one. Okay, so if the antibody isn't present, it's going to stop the virus from being able to agglutinate the red blood cells. Diagrams are always easier than words. Here's what it looks like. Right. So we have a known virus, right? We're adding this virus and we're adding red blood cells. If there's no antibodies, the virus is going to cause hemagglutination. It's going to make a big, gigantic mess. And those red blood cells are not going to be able to settle down to the bottom of the well. And so it'll look diffuse. Doing that same thing, if the patient's serum had antibodies, it's going to bind to the virus, freeing the red blood cells. They can then settle down at the end, at the bottom of the tube, and form a nice tight button. Okay, so with hemagglutination inhibition, detecting the antibodies, a button is a positive. Okay, the blood cell settling down to the bottom is positive. That means there was antibodies there. That's important to remember because there's a hemagglutination test, which is going to be the opposite. So for hemagglutination inhibition, a button is positive. I already mentioned this in that diagram. So standard amount of red blood cells, serial dilution of your patient's serum, standard amount of virus, mix them together, see what happens. So we have diffuse up here, a button down at the bottom. So again, diffuse means there was no antibodies there. It's a negative result. A button means there was antibodies and it's a positive result. See that positive control, you have buttons, negative control, no buttons. Right. And then we can do a serial dilution of the patient's sample. the last dilution in which you have a button is your endpoint titer, and that's how you would report it out. Right, so if we have patient A here along the top, is this positive or negative? Does the patient have antibodies? Zero dilution. Cross. Patient have any antibodies whatsoever? No, there's no button in that row, right? These last two are the details. They're only 1 through 10, right? There's no button. So this test result is negative. Patient has no antibodies. Right? From a technologist, lab worker perspective, you can call this test negative. From a clinical perspective, is this good or bad? Okay, or vote for bad. Why?
You don't have antibodies, you can protect yourself.
Okay, so that's one option. Could also be good. Right? What if this was a test for Ebola? I don't really want to have Ebola antibodies, to be perfectly honest with you, right? That means you had to have been exposed to Ebola. That's not such a great thing. Right, so when it's to highlight that we tend to look at tests in a vacuum of it's either positive or negative. But it's actually important to know what you're doing and what the test is for to decide whether it's actually a good thing or a bad thing. Right? If this is a test for a patient with Ebola, not having any antibodies is actually good. They weren't ever infected with Ebola. If this was a test for that was IgG for varicella zoster virus, I would agree this is a bad result. Right? Patient doesn't have any antibodies to VZV. That means they're susceptible to infection. And most of us, this were VZV IgG, we have some sort of a titer because we've either been infected. Most of you guys may have had the vaccine. I'm a wee bit too old to have gotten the vaccine. I got the good old fashioned way, right? But we all would have had antibodies. So context is everything with your test as to whether a negative or positive is good or bad. Okay? Just seeing it like this, you don't know. You just know that A is negative. Okay. How would we be reporting out B? Okay, they're positive. What about their titer? Right, 1 to 128. Right, that's the last dilution in which you have a positive result. Okay, they're negative at 1 to 256. Right, they might actually be positive somewhere in between here, but we don't test that. So C is 1 to 8, D is 1 to 4. Okay, if D was my acute phase antibody test, and C was my convalescent stage, would we say I had this disease? I have two choices. Yay or nay? What was the rule for being considered positive? Okay, so I need a fourfold dilution. Got 1 to 4 as my acute, 1 to 8 as my convalescent. Is that fourfold? So by this test, we would not say I had this virus, right? I've only got a twofold change. We need a fourfold. Does that mean I did not have this virus? No, serology is not perfect, right? But by the definition of what you need, a fourfold increase, I did not meet that. If D and C were my samples, right? If D and B were my two samples, I think that's more than a fourfold increase. Does that make sense to everybody? And I can tell you, you will probably never get an acute and a convalescent stage sample. It hardly ever happens. And technically, they actually have to be tested at the same time, not like the acute today, and then 4 weeks actually test the convalescent. You actually need to collect the acute and hold it to get the convalescent. So I can tell you, there's maybe a handful of times a year that happens. All right, so moving on to more fancy tests, the enzyme linked immunoglobulin assays. These are the ones that you're more likely to be doing than the hemocentration. Right, typically done in a microtiter well plate. Here again, if we're looking for antibodies in the patient, we're going to bind antigen, a known antigen, to the surface of the plate. Add the patient's serum, wash it away. Complex was formed. We then add a secondary antibody that recognizes the human antibody. Wash it away. It's conjugated to an enzyme. We add a substrate, and then we get a color change. That color change may be visible light, or it may not be visible light, but there's some sort of a change. Okay. So typically, the plates are read with a spectrophotometer, using the optical density. Because every day is going to be slightly different, you usually run some sort of a control, oftentimes a standard curve, so a few different solutions. And this is how you can get IgM and IgG done. This is what it looks like for antibodies. So we have our immobilized antigen. We add the human anti-antigen. So when you talk about antibodies, we have before, there's two different things. There's the species in which it was produced and the target that it attacks. Right? So if I say human anti-flu, that's a human produced antibody against the influenza virus. Right? Human anti-flu. Maybe that's what this is here. We wash away anything that was not bound. We add our secondary antibody that's conjugated to an enzyme, a reporter molecule. Right, this is oftentimes mouse anti-human. And so antibodies produced in a mouse directed towards human antibodies. Then add a substrate and look for the color change. For antigens, the principle is very similar. Bind an antibody to the surface, add the antigen. We then add a secondary antibody, but this time it's recognizing a separate isotope on the antigen. And so it's recognizing a different site on that antigen, but otherwise the principle is the same. It has a conjugated reporter molecule or enzyme, add a substrate, get a color change. Right, there's a variety of instruments that can do this. You can get yourself a spectrophotometer, do it all manually, incubate it all manually, and then take off the spectrophotometer. Or you can get one of these instruments that will pretty much do everything for you. Here's one of them. It's one of our old technologies. Here is the Abbott validity instruments that if you are rotating in our toxicology lab, see these guys. All different methods. Chemiluminescence is the one here, so it's not visible light, but same principles. Again, most laboratories, if you're doing some sort of analysis, you're using some type of instrument like this. You've all, I'm sure, done a lateral flow assay, probably at home at some point. Right, these are COVID tests. Now, flu is also available over the counter. Your pregnancy tests work very similarly. So, all basically, again, an antibody-antigen interaction. Here, the antibody, it's usually you're looking for antigens most of the time, but that helps for COVID. You do have ones that are for antibodies. And your antibody would be painted onto this strip of paper. You add your sample in a buffer, goes up. If the antigen and antibody interacts, you get a color change, and it looks like a line on there. Right? Pretty common. And typically, the at-home ones, of course, are qualitative. Most of the time, these are qualitative tests, but lateral flow assays can be used quantitatively. And for our cryptococcal antigen, we have a lateral flow assay that we do a quantitation off of. So it is possible to do quantitation with the lateral flows. Just the at-home versions that most people are used to are just quantitative from. Right. A Western blot is an old technique. This one, it's an adaptation of the enzyme immunoassay. It starts with an electrophoretic event. So on a gel, you add your sample of interest. It's coated so that all of the proteins have the same molecular charge. You then run a current through that gel, and the proteins will move through that gel based on their molecular weight. So larger proteins will move slower, smaller proteins will move faster. You then transfer those proteins to a piece of paper, nitrocellulose or PVDF. Again, using an electric current, this time going perpendicular to the gel. So you transfer them from here over to there. And then you can do antibody antigen interactions that way. This is what it looks like. And so we would have run a gel, transferred it to these strips of paper. And now we have protein antigens lining the paper. Then larger molecules up at the top, smaller molecules at the bottom. We block it with something. Research lab, we always use milk. Just to kind of cover up all these empty spaces so we didn't get any non-specific binding. Still done in the laboratory today. There's some sort of a blocking event. Then add your patient's sample. If the antibodies are present, they'll bind. And then depending on the test, you can have a secondary antibody, get a color change and see those lines. Research world, use radioactive isotopes, which are really fun, so glow in the dark. But just depends in the lab. It's going to be a secondary enzyme, just like those ELISAs, to get a color change. Okay. And depending on the test, you may have an algorithm for whether or not this is positive. Like maybe I need 5, for Lyme. Right, this is commonly done in HIV. Are the two most common where you use Western blot. Right, for Lyme, IgG positive, you need 5 out of the 10 bands. For IgM, you need 2 out of the 3 bands that are effective. Right. Realistically, nowadays, even though this is, you call it a Western blot's actually the whole technique going from electrophoresis on the gel to this. We don't usually do gels anymore. We buy these strips from the manufacturer, so all we have to do is add patient serum. You may hear the term immunoblot. Technically, since we're not going through the whole process, these are immunoblots, not Western blots. Most people use the terms interchangeably, but just so you're aware, you may hear the term immunoblot. It's referring to the same thing as a Western blot. Indirect antibody assays or indirect fluorescent assays, IFAs. And here we are looking for typically antibodies in the patient sample. So we're moving into kind of more old school techniques of using microscopy. We're going to put a known infected cell onto a slide. Influenza A. These cells are infected with influenza A. We're going to add the patient's serum. The antibodies bind. We can then add a secondary antibody. In this case, instead of being conjugated to an enzyme, it's going to be conjugated to a fluorescent molecule. Most of the time, that's going to be fluorescein isothiocyanate, which is also called FITC, and you get nice apple green color on the slide when you look at it under a fluorescent microscope. In theory, you can do titers with this method. Most commonly, though, it's just a yay or an nay. Very rarely will you see titers actually being performed, but it is certainly possible to do so. And here's what it looked like. Great, our known sample on the slide. Primary antibody for the human, secondary antibody that recognizes that one with the FITC attached. And you can see the nice apple green color in the fluorescent microscope. Typically, rhodamine is also added, which will stain the cells red, largely just to give you a good background. or something, right? So here we can see the red cells in there surrounded by the of this particular pathogen. And fluorescencing, because it's really sensitive, dust fluoresces. So you could see dust in the microscope. The antibody may non-specifically bind to some things, so you may see that as well. Right down here, you see some green. It's not the apple green that we see up here, which is one indication that it's just junk. But it also helps that I don't have a red cell that's attached to them, making me believe that, man, this is just background staining, right? If I didn't have any of these other cells positive, I would just call this junk, right? It's negative. We've talked about complement fixation before with the molds, right? And the same thing here with viruses. Right, so complement interacting with the antibody antigen complex, and whether or not it will lyse red blood cells. We have serum with or without antibodies. We add the antigen of interest, as well as complement and red blood cells. If the antigen is, or sorry, if the antibody is present, it's going to interact with that complement and the antigen and protect the red blood cells. They then get to settle down at the bottom and make a button. Like there's no lysis, so we see a button. That's reactive, it's a positive reaction because there was antibodies present. If there's no antibodies, the complement and antigen interact with the red blood cell and lyse them. So you get a diffuse red pigment solution. Okay? That is non-reactive because there was no antibodies present. So a lot of the principles that we talked about with antibody detection can also be used with antigens. There's some slight differences for some of the tests. And so direct agglutination versus the hemagglutination inhibition. And then there's direct immunofluorescence as opposed to indirect immunofluorescence. And so viral hemagglutination is basically the opposite of that neutralization for the inhibition assay. So we have a virus, this time we're adding the virus, or we have the virus present, hopefully, or not, that's what we're testing. We add our red blood cells and we look for hemagglutination. In this case, if we're not looking for antibodies, we don't care about antibodies now. If the virus was present, it's going to agglutinate the red blood cells and you get that diffuse pigment because they can't settle in the bottom. If the virus was not there, the red blood cells are protected, they're saved, they settle down at the bottom and make a button. Okay? So for the direct assay, the agglutination assay, a positive is diffuse pigment. If you're doing the neutralization assay, also called the hemagglutination inhibition assay, positive is a button. Okay. If that's confusing, please go back to the diagram and work it out. Okay. With all of these, you just need to look at what you're adding and what you're looking for. Okay. And you should be able to follow that process and determine, what should my end result be based on what I'm doing. Again, the viruses that you're most likely going to be used, influenza is the most common one. Parainfluenza as well. All right, here's what we have. So if this were direct agglutination, and the ones in this green box, we've got a button, which means what? This is direct agglutination.
Negative.
Good. See some confused faces. You all get that? The direct agglutination. Viruses not there. Cells get to settle down at the bottom, make a button. Right. How about in the black box? Positive or negative? Positive. Right? You don't see a button. So the virus was there, it gummed up the system, and the cells don't cell to the body. Right. This would be the opposite if we were looking for antibodies with the hemagglutination inhibition assay. Direct immunofluorescence and looking for antibody antigen interactions. This time, instead of having a known infected cell and adding the patient's serum, we're going to add a patient's sample to the slide and then add a known antibody to it. That antibody is going to be tagged again with a fluorescent molecule, again, usually FIT-C. Rhodamine, as mentioned, is usually the counter stain to detect the cells, and we're going to look and see what we have. It looks identical to the indirect fluorescent assay. Unless you are told what it is or what the samples are, you can't tell the difference between an IFA and a DFA. Right? They look identical. Typically, IFA is going to be used if you're looking for antibodies in a patient's serum. DFA is going to be used if you want to directly detect an infected cell. Right. Here's what that looks like. So really the big difference between DFA and IFA is whether or not there's multiple antibodies. With DFA, there's one antibody. Right? I'm looking for a specific antigen. I have a known antibody that I bind to. With indirect staining, I have a known antigen, an unknown patient sample, and then a known secondary antibody. But the big difference is IFA has 2 antibodies, DFA has 2. Monitoring disease progression, most commonly, especially for things like HIV or HCV, HBV, you're going to do viral load assays. So doing nucleic acid detection and looking how much virus there is. See with treatments, you want to see those numbers go down, preferably the non-detectable. Those go down. If they go up, that means either the patient is non-compliant with their treatment or the treatment is not working. For all those viruses, you can also do genotyping. And so it's kind of monitoring the virus, but you're looking to see what is the virus susceptible to so that you can tailor treatment better. Most commonly used with HIV, but it's also useful with HCV and HBV as well, hepatitis C and hepatitis B. But for us, we usually see it with HIV more commonly. Gives us the mutations. Those mutations then can be associated with resistance to certain antiviral agents, and the doctor can modify to get to things that are susceptible. It's also possible to use antigen titers. Right? In syphilis, the RPR, monitoring RPR goes a good way. Also, fungal, when we talk about histoplasma, you can monitor the antigens. All right, obviously the treatment been going down. Not commonly done with viruses, usually done with other pathogens, instead of syphilis or fungal infections. But again, it's a serology-based method. Sort of not thinking in the same way. That's the actual pathogen or antibodies to the pathogen. For something like HIV, we can monitor disease progression with flow cytometry, looking at the CD4 cells. Right, so still looking at antibody antigen interactions because we're using an antibody to CD4 cells with flow cytometry. Right? It is inversely affected by the virus. So as the viral load goes up, the CD4 cell count will go down, and vice versa. As the virus gets under control, the CD4 count hopefully starts to normalize, goes back up. So that is a way to monitor HIV progression without actually detecting the virus itself. And more of these will be discussed when you get to the actual lectures on those particular viruses and other molecular techniques. All right, the limitations of viral serology, we've already mentioned quite a few of them. One, there's a lot of cross-reactivity. So if we think about HIV testing, that screen is a very sensitive method detecting HIV. If you have a really highly sensitive method, you usually take a hit on specificity. So there's a lot of cross-reactivity with things, many of the things we aren't even aware of, will cause a positive reaction. Okay, so serology is not perfect. It's prone to false positives and false negatives. Right? It's important to understand the epidemiology of the pathogen you're trying to detect with serology. If IgG does not show up until month number 3, if the patient was potentially infected yesterday, is it worth doing IgG testing? Now, the patient's going to be negative, but that doesn't mean they're healthy. Right? So you have to know the disease course. And if we want to use IgM, but the patient was infected 6 months ago, is that likely to be useful? Probably not. The IgM is probably gone by now. Right? The same is true with molecular testing using nucleic acid amplification techniques. Right? There's limitations there based on the epidemiology of the infection. It's generally more sensitive than serology, particularly early on. So in the acute phase of the illness, you usually want to use a NAT-based method. After about a month or so, again, depends on the pathogen, but after about a month or so, you're probably going to be better off doing IgG testing because the pathogen is likely to be gone. And we already mentioned the timing with acute and convalescent and the patient already resolving, good or bad, before you will actually know those results. Any questions related to serology? Just be careful with some of the testing again. Helpful to kind of work out what am I actually looking for? What am I adding to it to be able to do that? Will help determine what reaction is positive versus negative. Always helps to diagram it out if you have to.